Hi, everyone. Welcome to join us for the webinar. I'm Juan Liang from R&D department at GemPharmatech. Today, I will share the autoimmune disease model that were generated using the mice with humanized immune systems. Before we start the webinar, I would like to give a brief introduction about our company. GemPharmatech is a global biotech company that provides a one-stop solution for biomedical research using genetically engineered mice model. We are the world's leading mouse model repository. So far, we have generated over 25,000 models, and we have the capacity to generate over 600 models annually. And we can also perform over 500 preclinical testing projects. We have set up 6 auction sites across China and US, and the US site is set up at San Diego. So let's back to the autoimmune disease model. So what is autoimmune disease? Autoimmune disease occurs when the immune system mistakenly attacks its own body tissues. It could happen localized. It can affect in certain parts of the body, like blood, brain, nerves, lungs, skin, and bones, muscles, and also the other organs. It could also happen systemically, attacking multiple organs or body systems, like the SLE syndromes. Also, the mechanism that causes autoimmunity is complicated, but we all know that the immune system disorder is the main modulators that cause the autoimmune disease happened. The innate immune systems like the dendritic cells, the neutrophil cells, NK cells, complement macrophage can contribute to the development of autoimmune disease. The adaptive systems like the T cells, B cells also contribute to the pathogenesis of autoimmune disease. It's like the innate immune cells will secrete the cytokines that will activate the adaptive systems. So the active systems like the B cells will generate the autoantibodies that against self-antigens. The T active cells can also against directly to the self-antigens. So the interaction and the disorder of the innate and adaptive cells could both contribute to the autoimmune disease development. Here is a list of the autoimmune disease model we have generated at GemPharmatech. We have generated the immune disease model related to skin, like psoriasis, atopic dermatitis, and also the digestive system-related autoimmune disease model, like UCCD, and also the other organ-related disease like arthritis, nephritis, asthma, multiple sclerosis, and also the SLE. For each immune disease model, it may trigger and driven by different mechanisms, as indicated by the different color of the dots. Some models may be driven by T cell activation. Some models may have the B cell activation, and the innate cells and also complement factors also contribute to the disease model development. So as indicated in this list, we know that the autoimmune disease is happened by the interaction of various immune cells. So it's very important to get the model to have the systems that could involve all the immune cells, especially if we would like to study the mechanism and also to test the drugs that could reflect the clinical drug response. We should have the mice model, the preclinical model that could involve all the human immune cells. So here is our a solution to generate such animal models. Based on our study platform, we have generated the reconstitution mouse model. In our reconstitution mouse model, we could develop a human myeloid cells, NK cells, DC cells, B cells, T cells, and also the other immunocells. We can expression the human cytokines in the immunodeficient mice to get the immunodeficient mice expression human cytokines. So those different cytokines could help the different immune cell reconstitution in the mice. Here is the figures that show what kind of cytokines that could support the different immune cell differentiation. As indicated here, the IL-3, GM-CSF, SCSF could support the differentiation of common myeloid progenitors, like the mast cell, like the neutrophil cell, intestinal cell, monocyte. And also the IL-2 and IL-15 cytokines could support the common lymphoid progenitors, like the NK cells and also T cells. So we expression the different human cytokines in the immunodeficient NCG mice to help to support the different immune cell differentiation. As shown here, we have generated this NCGM mouse strain. This strain can expression human SCF, GM-CSF, and IL-3. Those three cytokines can support the myeloid cell differentiation and establish the monocyte, macrophage, and also DC cells in this mouse strain. And we also generate the IL-15 and IL-2 humanized mice model in the NCG deficient mice. So in this mice strain, this mice could help to generate NK cells. And when we breed in these two mouse strain together, we can get this NCGM IL-15 mouse strain. This strain can have the cytokines that could support myeloid cells development and also the NK cell development. Here is a comparison of the next generation of NCG mice we have generated at GemPharmatech. As we mentioned, when the NCG mice expression human IL-15, this mice strain could support the human T cell, B cell, and NK cells, but with very little population of myeloid cell. And also in this IL-2 expression NCG mice strain, it could help the human NK cell development. Today I would like to share some data about the NCGM mice strain. In this mice strain, we expression the SCF, GM-CSF, and IL-3. Those human cytokines can help to develop human T cells, B cells, and also myeloid cells in these mice. Since these mice have various human immune cells, so we can use this mice as the tools to induce the autoimmune disease in this model. So these mice could be very valuable models for us to study the human immune systems and the contribution of human immune systems for the development of autoimmune disease, and it will also help us to evaluate the drugs that targeting for human targets to evaluate the drug efficacy. Before we start to introduce the autoimmune disease model, I would like to give some information about this immune cell reconstitution mouse, the HSC-NCG mice. As indicated here, the red curve is NCGM mice and the black one is NCG mice. As we mentioned, both these immunodeficient mice could support human immune cell reconstitution. So we transplant the human HSC to the immunodeficient mice and to reconstitution human immune cells. As indicated here, the NCGM mice could re-establish the human CD3 positive cell, CD19 positive B cell, and also the NK cell, monocyte cell. And it can also reconstitution the T cell subpopulation like CD4 positive T cells, CD8 positive T cells, and also the Treg cells. And also once expression of the 3 human cytokines in this NCG mice, the NCGM mice could reconstitution high levels of CD3 positive cell and also the other immune cell population compared with NCG mice. Also, maybe we could find the B cell ratio might be a little bit decreased compared to the NCG mice. But when we look at the B cell count, we still have increased B cell count in the NCGM compared with NCG, in the NCGM mice compared with the NCG mice, which the data I have not shown you here. And also those, when we transplantation of the HSC into the NCGM mice, they could also help the spleen and cerebral development. So this HSC NCG M mice could be used to study the immuno-oncology, the autoimmune study. They could also be used as the infection models and could also be used for the safety evaluation. Today, I would like to share some data that we already generated for the autoimmune disease, which has been used in this HSC NCG mice. We will introduce 3 autoimmune disease models that has been generated in the HSC NCGM mice. One is arthritis, which is induced by CFA. The other one is SLE model that was introduced by TLR7. The last one is DSS, is the IBD model, which is induced by DSS. So the first one is a CFA-induced arthritis model in HSC NCGM mice. As I mentioned, first we will establish human immune system in this NCGM mice with transplantation of the human HSC into the NCGM mice. After around 12 to 40 weeks, we could reconstitution human immune cells, including T cell, B cell, myeloid cells, and also the other innate immune cells. And then we injection CFA into the mice to induce arthritis. As indicated by this representative image, we could observe the feet swelling in this model. And also we could observe the increased arthritis score in this modeling group compared with the non-treatment group. When we look at the detail of the arthritis score, we could observe the increased thickness of the pole and also increased swelling of the pole. And we also found the increased secretion of IL-6 in the modeling group that indicate the macrophage have been activated by in our arthritis model. And in the histopathological study, we performed the HE study and also the TRAP studies. From these figures, we could find that the modeling group exhibited heightened inflammation levels and also the bone erosion. We also observed the other bone damage in this modeling group. We also very curious about the immuno cell population which have been activated in our model. So we performed the HSC study. In the modeling group, we could find the increased level of the CD3 positive marker and also PD1 positive marker, as well as the CD68 positive marker. That indicate the increased infiltration of T cells and also the macrophage in this arthritis model. Then we performed the drug efficacy study in this HSC NCGM arthritis model. First, we evaluate the TNF-alpha antibody efficacy using this model. We already know that TNF-alpha contribute to the development of arthritis development. And also the antibody of TNF-alpha, the adalimumab has already approved by FDA for the clinical arthritis patient treatment. So in this model, we performed a study to evaluate this drug efficacy. First, we established this immunoreconstitution model with transplantation of human HSC around 10 to 40 weeks after the immune system reconstitution. We treat the mice with the CFA to induce arthritis. And during the study period, we will score arthritis, measurement the limb thickness, and also measure the claw swelling. And we around about 2 weeks, we will do the 10-point analysis. So here is the body weight change during the study period. And compared to disease model group, the TNF-alpha antibodies and also the IL-6 antibodies could alleviate the body loss. When we look at the arthritis score, we could find that the TNF-alpha antibodies could significantly decrease the claw thickness and also the claw thickness to weight ratio, but the IL-6 antibodies exhibited no obvious efficacy. And also, when we look at the swollen scores, the TNF-alpha antibodies also showed the significant alleviation effect to reduce the swollen scores. The comprehensive arthritis score also indicates that the TNF-alpha antibodies could reduce the arthritis score, but the IL-6 antibodies exhibited no obvious efficacy. Similar results was also observed in the survival rate. This TNF-alpha antibodies, as indicated in this blue curve, could improve the survival rate. And also, when we look at the IL-6 levels released by macrophage, the TNF-alpha antibodies could also reduce the IL-6 release compared with the modeling group. We also observed the immune cell population after the drug treatment. And in this study, it seems that in the peripheral blood, the T cells could be activated in the modeling group. And when we treat the mice with the TNF-alpha antibody, the treatment group could reduce the T cell population. Here is also the similar results that present the cell population counts. Also, the T cell counts were increased in the modeling group, and TNF-alpha antibody treatment could reduction the T cell population. So this human immunoreconstitution model could be used to generate the arthritis model. This model could develop of fully functional human immune systems, and it could also showing the RA syndrome that very similar to clinical patients. In this model, we could evaluate the FDA-approved drugs, and the model also responds to the drug treatment, which is very similar to the response that already observed in clinical patients. As indicated here, we can transplantation the HSC into the NCGM mice to reconstitution human immune systems, and we induce CFA to induce the syndrome of arthritis, and we do the arthritis score evaluation, and also we can evaluate the cytokine release. This model has been accepted in the ACR conference as a poster presentation in last year. And in this model, we also perform another drug efficacy study. In this drug study, we evaluate the IL-17 antibody efficacy. IL-17 is also the main factors that contribute to the arthritis development. So similarly to the previous study, we established this arthritis model in the HSC NCG mice and evaluate the IL-17 antibody drug efficacy. The data shown here that we could observe that as indicated in this curve, the IL-17 antibodies could reduce the claw swelling level and also could reduce the claw thickness. When we look at the comprehensive arthritis score, the IL-17 antibodies could also reduce the arthritis score, which is also very similar to the drug response that already observed in clinical patients. So the next autoimmune models I'd like to introduce is the TL7 HSC NCG mice. We use this method to establish the SLE disease model in these mice. Here is the mechanism of the SLE, how the SLE happened. Although the pathogenesis of SLE is very complicated, but the major driven is the disorder of the immune systems, especially the activation of PDC cells. In the clinical patient, the some driven factors will activate the PDC cells, so the PDC cells can release the type 1 cytokine, type 1 interferon cytokine. The type 1 interferon cytokine can activate the APC cells. So the APC cells can activation of T cells. Then the T cells can activate the B cells. The autoreactive B cells can release auto antibodies that targeting for the for the organ and tissues. So the SLE development. So based on this mechanism that involved in the development of SLE, we try to mimic the similar mechanism that in our mouse model to reflect the phenotype and also the mechanism that happens in patients. In this model, we transplant the human HSC into the NCGM mice, and then we use a TR7 to activation the PDC cells. TR7 and also the TR9 are expressions in the PDC. So when we use a TR7 agonist, we can activate the PDC cells. So the PDC cells can interact with the other immune cells like the APC, T cells, B cells, so we can mimic the manifestation of the patients in our disease models. Usually, for this kind of SLE models, we can use two different methods to generate this SLE model. One method is we can use the PBMC transplantation, human PBMC transplantation to the immunodeficient mice. Then we can get the human SLE model. But in this mice, since we only transplantation the PBMC into the mice, so this mice will only have the T cells and some B cell functions, but no the innate cells like the PDC cell or the other APC cells involved in the model. If we use the HSC transplantation into the NCG deficient mice, we can establish the innate immune cell like the PDC cell, myeloid cell, and also we can have the T cells and B cells. So in our study, we use this method to generate the SLE models. And we can also use preclinical to induce SLE in these mice. But when we use preclinical, usually the preclinical can only activate activation of myeloid cells, but not PDC cells. So in our study, we use a TR7 agonist to active PDC cells, which will mimic the mechanism that have been observed in clinical patients. Here is a study. Here is a study timeline for the drug efficacy. First, we also need to reconstitution human HSC in these mice. After 10 to 40 weeks reconstitution of human immunocells, we treat the mice when the TR7 for around 6 weeks. And then we could perform the phenotype validation and also the drug efficacy studies. During the study period, we will do the effects analysis to study the immunocell population, and also we will detect the IgG and also the anti-double-stranding levels. Those two parameters are the important indicators for the SLE. And also in the end time point, we will also do the signal study and also the histopathological studies to evaluate the drug efficacy. This figure showing the immunocell population in our model. As indicated here, showing here, the TR agonist treatment could induce the immunocell differentiation compared to the vehicle treatment group. So we can observe the increased CD3 positive cell population and also increased level of CD19 positive B cell population increased when the TR7 agonist treatment. And also, as I mentioned, the PDC cell is very important for the development of SLE. So in our mouse model, we also observed the increased level of PDC cell after we treat the mice with the TR7 agonist. And also in this treatment group, after treatment with TR7, we could observe the increased level of anti-double-stranded DNA and also IgG level. Those serum parameters, anti-double-stranded DNA and also IgG is very important indicators for the development of SLE. And also, we observed the rash on face and back, hair loss on neck and back in our mouse model. And we also observed the dysfunction of kidney, like we observed the deposition of the IgG immunocomplex in the kidney. And then we conducted the drug efficacy study in this HSC NCGM SLE models. In this study, we mainly focus on the drugs that targeting for the B cells. One drug we test is the belimumab. Belimumab is the antibody targeting for BAFF, and BAFF is the important markers that expression in the B cells. And another drug we tested here is the rituximab. This antibody is CD20 antibodies. CD20 is also the B cell markers. So here is our drug efficacy figures. From this figure, we can observe that those two curves, the yellow one and also this blue one, are the efficacy group, are the drug treatment group. And from the data, we can see the drug treatment could improve survival rate. And also, when we look at the serum parameters, the IgG and anti-double-stranding levels, the drug treatment group, as indicated here, they can also reduce the IgG level and also anti-double-stranding levels. So those two drugs showing the benefit efficacy in this model. And in this model, we also evaluate the bispecific antibody drug efficacy. We tested two drugs here. One is a CD3 CD19 positive. One is a CD3 CD19 bispecific antibody. The other is a CD3 BCMA bispecific antibody. Both drugs are targeting for the B cells. And as shown here, after 20 days treatment, we could observe the both the bispecific antibodies could reduce the human anti-double-stranding levels, and also those two bispecific antibody technical B cells could reduce the lymphocyte cell population in this model. Here is individual data of each mice that responds to the drug treatment. Although maybe not every mice responds well to the drug treatment, but we can really have some data that with the treatment of the bispecific antibody, they can really reduce the IgG levels. And here we show the FACS data of the immunocell population after 13 days of treatment. It's very interestingly, only the CD3, CD19 bispecific antibodies could reduce CD19 ratios as indicated here after 30 days treatment. But when we look at the cell counts in the blood, both the bispecific antibody treatment could reduce the CD19 positive B cells levels. And similar results was also observed at 24 days treatment. So also the bispecific antibody treatment could reduce the B cell level in the percentage. And also for the cell count, all the bispecific antibody could reduce B cell level. And we also curious about the B cell subpopulation when after treatment with the drugs. So we analyze the B cell subpopulation like plasma cell and also the MZ B cells. And we could observe that for the percentage analysis, the MZ B cells could be reduced by the drug treatment. And when we look at the cell number after treatment, all the B cell subpopulation we have detected here, all showing the decrease, all have been decreased by drug treatment. So the last autoimmune disease model is a DSS-induced colitis model in HSC NCGM mice. Also, when we establish this model, we need to reconstitution human immunocells for around 10 to 40 weeks. And then we add the DSS into the drink water and administration to the mice. After around 2 weeks, and during the administration, we will treat the mice with the, and after around 2 weeks treatment, we will do the end time point analysis, including FACS analysis, measure the colon length, and also perform the pathological study. During the induction period, we will scoring the disease activity index and also measure the weight loss. Here is the data showing the model development. As indicated here, we treat the mice with the DSS as indicated in the yellow, blue, and also green lines. The mice are showing the body weight decrease. And also when we look at the DI score, the disease activity index score, we could also observe the increased DI score when we treat the mice with DSS. And in the endpoint, we measure the colon length. We could find the decreased colon length when we treat the mice with DSS, which is also the indicators which suggest that these mice develop colitis. And we also, and here is also the score of the colon tissues. the treatment when the DSS could also increase of the other scores like the immunocell infiltration in the colon tissue. And here is the pathological study we performed in this disease model. And from this histological studies, we could observe the infiltration and also the epithelium structure damage in colon after we treat the mice with DSS in these immunodeficient mice. And also, we would like to know what kind of immunocells have been activated in these models. So we performed the RHC study and detect the CD3 positive T cells. As indicated here in the modeling group, we could observe the increased infiltration of T cells. So here is the summary about the immunoreconstitution model we have generated at GemPharmatech. With transplantation of the human HSC into the mice, we could generate various immunocells like the T cell, B cell, DC cell, monocyte, neutrophil, macrophage, and also T cell, B cells. And then we establish the autoimmune disease based on this human immunocell reconstitution mice. As shown here, the CFA-induced arthritis model is developed by activation of innate cells and also T cells. The SLE model induced by TL7 is activation by T cells and also B cells. For this colitis model that induced by DSS, it was driven by the innate cells and also the T cells. So this immunoreconstitution autoimmune disease model could be used for the study of monotherapy, combo therapy, bispecific antibody. Since this model already has the human immunocells, so we can perform the study, perform the drug efficacy study that specifically recognize human markers. So in this model, we can perform the monocyte, composite, and also the bispecific antibody efficacy study. And more importantly, since these mice are immunodeficient mice, we can also transplant the therapeutic cells to these mice to evaluate the cell therapy efficacy for the autoimmune disease treatment. And also, besides this model we have introduced, we also host another webinar that is about the SLE model development and also the drug efficacy study. Our audience can visit our website to get this webinar results. So here is the model that I would like to share. And now I'm ready to take the questions. We have the question here. Which immunocell population was activated in the arthritis model? So in our arthritis model, we have detected the markers of macrophage and also the T cells like CD68 positive cells and also the CD3 positive cells. So that means we could activation the macrophage and also the T cells. And we also evaluate the drugs that detecting for macrophage and T cells like the TNF-alpha and IL-17 antibodies. Both drugs showing the benefit effect in this disease model. So that also means the macrophage and T cells were activated in this model. Yes, you can record this webinar, and we can also share the webinar to our audience. Do you have autoimmune disease model for cell therapy? Are these mice probably models for cell therapy evaluation? Yes. As I mentioned, since the model we used is immunodeficient mice, so these mice could be probably for transfer the therapy cells into the mice, and we can actually we have some project that that we are working on to evaluate the cell therapy efficacy using this model. We have done this kind of study to evaluate the CAR-NK and also CAR T cell efficacy in the SLE models, and we could find this this CAR-NK and also CAR T cells could improve the cell survival. It could improve the mice survival in these studies, and also we could observe the decreased level of the double-stranded DNA. So this immunoreconstitution mice model could be used to evaluate therapy that for the autoimmune disease treatment. So we have the question here, what are the readouts in your SLE model? So in the immunoreconstitution SLE model, we could observe the increased level of double-stranded DNA in serum and also increased IgG level in the serum. In some case, we can also observe the kidney dysfunction like the IgG complex, like the IgG immunocomplex deposition. But sometimes it might be based on the donor we have chosen for immunoreconstitution. So the basically readouts in our model is a serum IgG level and also double-stranded DNA level. We try to set up the MS model to test the cell therapy, but it's quite complicated for this model generation. Since for the MS model established, we need to have the fully functioned PDC cells to present the antigen to T cells and also B cells. So the B cell can generate the autoantibody to destroy the neuron cells. But in this kind of immunoreconstitution mice, it's quite difficult for the PDC cell to present the antigen to T cells. Usually, we need to culture PDC cell in vitro and preloading the antigen to PDC cells. So it's quite complicated. We are trying to work on this kind of models. Do you have pre-screened CD3 donors for the reconstitution to ensure high success rate? We have some donors that we already tested that could work well in the reconstitution model for autoimmune disease model generation. But sometimes if we try different indication, maybe we require different immunocell populations that to be established. Like in some autoimmune disease model, we would like to have high T cell population, and in some autoimmune disease model, we would like to have the increased B cell levels. So basically, it depends on what kind of indication we are looking at. So when we when we group the model, besides the CD33 positive cell, we can also look at the other cell population. If no more question, I think we can stop here. Thanks again for joining us for the webinar. Have a nice day.